This video is about external fertilization in soya regions. Developmental biology portion. So let's start. Recognition of sperm and egg. Chemoattraction of the sperm to the egg by soluble molecules secreted by the egg. Exocytosis of the sperm acrosomal vesicle and release of its enzymes. Binding of the sperm to the extracellular matrix, vitelline envelope or zona pellucida of the egg, passage of the sperm through its extracellular matrix, fusion of the egg and sperm cell membranes. Sometimes step 2 and 3 can be reversed, as in mammalian fertilization, and sperm binds to the extracellular matrix of the egg before releasing the contents of the acrosome. In sea urchin, sperm contacts jelly layer, acrosome reaction, digestion of jelly layer, binding to vitelline envelope, fusion of acrosomal process membrane and egg membrane. In case of mouse, sperm activated by female reproductive tract, sperm binds to zona pellucida, acrosome reaction, sperm lyses whole in zona, sperm and egg membrane fuse. Then sperm attraction. A small chemotactic peptide protein amino acid resact secreted from egg jelly of sea urchin A. punctata and bind with the extracellular side of the receptor present on sperm cell membrane. After binding, it activates latent guanylyl cyclase, causes sperm cell to make more cyclic GMP that activates a calcium channel, allowing the influx of calcium ions from the seawater into the sperm, activates sperm motility, thus providing a directional cues along the resact gradient towards the egg. A single resact molecule is able to provide direction for the sperm. Then the increase in cGMP and Ca2 plus activate both the mitochondrial ATP generating apparatus and the dynein ATPase that stimulate flagella movement in the sperm. In addition, the sperm sends the sap gradient by curving their tails, interspersing straight swimming with the turn to sense the environment. The binding of a single resact molecule may be enough to provide direction for the sperm, which swim up a concentration gradient of this compound until they reach the egg. This resact functions as a sperm attracting peptide as well as a sperm activating peptide. Acrosome reaction. A second interaction between sperm and egg jelly results in the acrosome reaction, where the sulfate containing polysaccharide binds to specific receptors located on the sperm cell membrane above acrosomal vesicle that has two components: the fusion of the acrosomal vesicle with the sperm cell membrane and exocytosis that result in release of the contents of the acrosomal vesicle, the extension of the acrosomal process. In Strongylocentrotus purpuratus, the acrosome reaction is initiated by a repeating polymer of fucose sulfate, and this sulfated polysaccharide binds to its receptor on the sperm. The receptor activates three sperm membrane proteins: a calcium transport channel that allows Ca2 plus to enter the sperm head, a sodium hydrogen exchanger that pumps sodium ions Na plus into the sperm as it pumps hydrogen ions H plus out, the phospholipase enzyme that makes another second messenger, the phospholipid inositol 1,4,5 triphosphate. IP3 is able to release Ca2 plus from inside the sperm, probably from within the acrosome itself. The elevated Ca2 plus level in a relatively basic cytoplasm triggers the fusion of the acrosomal membrane with the adjacent sperm cell membrane, releasing enzymes that can lyse a path through the egg jelly to the vitelline envelope. The second part of the acrosomal reaction involves the extension of the acrosomal process by the polymerization of globular actin molecules into actin filaments. The influx of Ca2 plus is thought to activate the protein Rho B in the acrosomal region and mid-piece of the sperm. This GTP binding protein helps organize the actin cytoskeleton in many types of cells and is thought to be active in polymerizing actin to make the acrosomal process. Then recognition of the egg extracellular coat, sperm adhesion to the egg surface. Another species specific recognition occurs by the acrosomal protein mediating called bindin located on acrosomal process. Species specific bindin receptors on the egg vitelline membrane. Sperm binding occurs on the egg surface vitelline envelope, where a limiting number of sperm binding sites are there in a species specific manner. ERB1 is a glycoprotein that is bindin receptor isolated from sea urchin eggs. Closely related species of sea urchins have divergent binding receptors, and eggs will adhere only to the binding of their own species. After having passed through the egg jelly coat, the spermatozoa encounter the vitelline envelope, which is a tough non-cellular layer present between the jelly coat and plasma membrane. The vitelline envelope is made up of glycoprotein and polysaccharide molecules. The spermatozoa digest their way through it with the help of acrosomal enzymes and trypsin-like activity, commonly called lysin. Then fusion of the egg and sperm membrane, egg cell membranes. Once the sperm has traveled to the egg, undergo the acrosome reaction, the fusion of the sperm cell membrane with the egg cell membrane of egg can begin. Sperm-egg fusion appeared to cause the polymerization of actin in the egg to form a fertilization cone. The actin from the gametes form a connection that widens the cytoplasmic bridge between the egg and sperm. The sperm nucleus and tail pass through the bridge. Fusion is an active process mediated by fusogenic proteins. Like in sea urchin, sperm bindin plays a second role as a fusogenic protein. So bindin contains a long stretch of hydrophobic amino acids near its amino terminus, and this region is able to fuse phospholipid vesicles under proper. Fusion is an active process mediated by fusogenic proteins. Like in sea urchin, sperm bindin plays a second role as a fusogenic protein. Bindin contains a long stretch of hydrophobic amino acids near its amino terminus, and this region is able to fuse phospholipid vesicles. Under proper ionic conditions, bindin can cause the sperm and egg membrane to fuse. The fast block to polyspermy. The entrance of multiple sperms, called polyspermy, lead to triploid set of chromosomes in organisms. The species have evolved ways to prevent the union of more than two haploid nuclei. The most common way is to prevent the entry of more than one sperm into the egg by a fast reaction accomplished by an electric change in the egg cell membrane. Within 1 to 2 seconds after the binding of the first sperm, the membrane potential shifts to positive level, about plus 20 MV. This change is caused by a small influx of Na plus into the egg. Sperm can fuse with membrane having a resting potential of minus 70 kV MV. They cannot fuse with membranes having a positive resting potential, so no more sperm can fuse to the egg. Fusogenic reaction of bindin will not function on a positively charged surface. If supply of sodium ions is not sufficient to cause the positive shift in membrane potential, polyspermy occurs. Then the slow block to polyspermy. The slow block of polyspermy. Cortical granule reaction, a slower mechanical shock to polyspermy that become active about a minute after the successful sperm egg fusion. In sea urchins and mammals, rise in C2 plus concentration responsible for the cortical granule reaction is not due to an influx of calcium into the egg, but comes from within the egg itself. A cortical granule undergo exocytosis. They release cortical granules, serine protease, an enzyme that cleaves the proteins linking the vitelline envelope to the cell membrane. Mucopolysaccharides released by the cortical granules form an osmotic gradient, causing water to enter and swell the space between the vitelline envelope and the cell membrane. The enzyme UDX1 in the form of cortical granule membrane catalyzes the formation of hydrogen peroxide H2O2, the substrate for soluble ovoperoxidase OVOP. OVOP and transglutaminase TG harden the vitelline envelope, now called the fertilization envelope. The fertilization envelope starts to form at the site of sperm membrane and continues its expansion around the egg. The fertilization envelope starts to form at the site of sperm membrane and continues its expansion around the egg. These cross-links allow the egg and early embryo to resist the shearing forces of the ocean's intertidal waves. As this is happening, a fourth set of cortical granule proteins, including hyaline, forms a coating around the egg. Calcium as the initiator of the cortical granule reaction. In sea urchins and mammals, upon fertilization, the calcium ions are released into the cortical cytoplasm of egg from internal store of the egg. In the high calcium environment, the cortical granule membrane fuse with the egg cell membrane, releasing their contents. The calcium ions do not merely diffuse across the egg from the point of sperm entry. Rather, the release of Ca2 plus starts at one end of the cell and proceeds actively to the other end, creating a wave of calcium across the egg. The entire release of Ca2 plus is complete within roughly 30 seconds. An activation of egg metabolism in sea urchins. The membrane phospholipid phosphatidylinositol 4,5-biphosphate PAP2 is split by enzyme phospholipase C, PLCC to yield IP3 and DAG. IP3 release Ca2 plus ions into cytoplasm from ER, and DAG activates PKC, which activates a protein that exchanges sodium ions for hydrogen ion, raising the pH of this egg. The IP3 formed at the site of sperm entry. So SRC proteins are found in the cortical cytoplasm of sea urchin that can form complex with PLC gamma. So heterotrimeric G protein in the cortex of the egg are activated by binding of the sperm bindin that activates SRC kinase that directly activate PLC gamma and Ca2 plus level elevated in the egg cytoplasm. So diffusible compounds from the sperm also release calcium directly to the ER upon contact with egg jelly, NADP. Concentration in sea urchin sperm increase that frees stored Ca2 plus from membrane vesicles. Effects of calcium. Early response. Calcium release activates a series of metabolic reactions that initiate embryonic development. One of these is the activation of the enzyme NAD plus kinase, which converts NAD plus to NADP plus. Since NADP plus or not NAD plus can be used as a coenzyme for lipid biosynthesis, such a conversion has important consequences for lipid metabolism and thus may be important in the construction of many new cell membranes required during cleavage. UDX1, the enzyme responsible for the reduction of oxygen to cross-link the fertilization envelope, is also NADPH dependent. Lastly, NADPH helps generate glutathione and oothiols, molecules that may be crucial scavengers of free radicals that could otherwise damage DNA of the egg and early embryo. And later response, resumption of protein and DNA synthesis. The rise in the concentration intracellular pH begins with a second influx of sodium ions. pH increase and Ca2 plus elevation act together to stimulate new DNA and protein synthesis. The wave of free Ca2 plus inactivates the enzyme MAP kinase, converting it from a phosphorylated active to an unphosphorylated inactive form, thus removing an inhibition on DNA synthesis. In sea urchin, the maternal mRNA include proteins such as histones, tubulins, actins, and morphogenetic factors that are used during early development. Release of translation inhibitor from mRNA. One of the mRNAs freed by the degradation of EIF4E binding protein is the message encoding cyclin B. The cyclin B protein combines with CDK1 cyclin to create mitosis promoting factor MRF, which is required to initiate cell division. Fusion of genetic materials. The mitochondrial genome is transmitted by the maternal parent. Inside the cell, the sperm nucleus undergoes transformation as it decondensed by the protein derived from the egg cytoplasm. Sperm decondensation initiated by the phosphorylation of the nuclear envelope lamin protein that begins when sperm contact with the glycoprotein in the egg jelly through CAMP pathway. The loosening of histones is replaced in sperm by other histones stored in the oocyte cytoplasm. After the sea urchin sperm enters the egg cytoplasm, the sperm nucleus separates from the tail and rotates 180 degrees so that the sperm centriole is between the developing male pronucleus and the egg pronucleus. The term sperm centriole then acts as a microtubule organizing center, expanding its own microtubules and integrating them with egg microtubules to form an aster. So microtubules extend throughout the egg and contact the female pronucleus, at which point the two pronuclei migrate toward each other. Their fusion forms the diploid zygote nucleus. DNA synthesis can begin either in the pronuclear stage or after the formation of the zygote nucleus. This depends on the level of Ca2 plus release earlier in fertilization.